Hello, today we're going to present an implantable biosensor system to continuously monitor troponin for early heart attack diagnosis. Every 40 seconds in the US, someone suffers from a heart attack, summing up to 805,000 per year. 20% of these are recurring heart attacks that happen within 5 years of the first one. And it is unpredictable when this will happen again until symptoms start to appear. If they ever do, as 1/5 of the heart attacks are silent, but damage still happens. There is no current way to prevent heart attacks besides medication and healthy lifestyle. So a fast diagnosis time is critical to avoid chronic heart tissue damage, which can affect patients' daily life and will experience short breath with any minimal activity if they're lucky to survive. Patients need to recognize a heart attack is happening in order to start diagnosis. And according to the Society of Cardiovascular Interventions, there are only 90 minutes since the onset of a heart attack to the detection for damage to be reversible. Patients sometimes takes up to 2 days to seek for help, and that's too late. So there's a need for diagnosing tool that detects early heart attacks onset and alerts the patients to seek for help before the damage is irreversible.
Heart attacks are considered acute events and are related to a blockage in the blood vessels preventing blood from reaching the heart or brain. They are sometimes occur when the arteries spasm. The main risk factors that contribute to the occurrence of such diseases are directly related to a person's lifestyle, which include poor diet, physical inactivity, alcohol use, tobacco consumption, and heredity. At the arteries level, the buildup of fat and cholesterol in the bloodstream leads to the formation of a plaque called atherosclerosis, which causes narrowing of blood vessels, resulting in less oxygen delivered to the heart. This atherosclerotic plaque may rupture, causing a clot formation that leads to a complete blockage rather than performing its main intended function, which is attempting to fix the damage. Before being phased out due to their lack of specificity for cardiac muscles, AST, LDH, and CK were used as cardiac biomarkers to identify myocardial ischemia. A specific and extremely sensitive marker can be linked to the detection of myocardial necrosis, known as troponin biomarkers, found in the cytoplasm of cardiac myocytes. The structure of troponin is altered upon calcium binding to the protein, which causes interactions between actin and myosin filaments, resulting in cardiac contractions, as seen in the image to the right. The protein complex is formed of troponin C, troponin I, and troponin T. Troponin I and troponin T are responsible for identifying cardiac muscle injury. During myocardial infarction, known as heart attack, cell membranes rupture, leading to the spill of intracellular contents to the extracellular space that will end up in the bloodstream. Cellular contents, including troponins, will be detected in the circulating blood when spilled in larger quantities. These levels are found in basal amounts in healthy individuals to be less than 0.04 ng per milliliters. Those levels start to elevate from 0.7 to 0.07 to 1.4 ng per milliliters within 2 to 3 hours of chest pain distinguished symptom. Those levels continue to rise, reaching 100 ng per milliliter within a 48-hour period. And the level is supposed to fall back within 4 to 10 days.
Current methods to diagnose heart attacks are EKGs, which is a painless procedure but only diagnoses major heart attacks, and troponin blood test, which is the gold standard due to its accuracy, but it may have false negative if run too early or may have to be run multiple times to keep monitoring. Both of them only take around 15 minutes to run. However, first, the patient needs to realize that they're having a heart attack, then go to the ER and wait till someone takes care of them. And this may take up hours, if not days. And tissue damage will be reversible. And that's why we propose a novel implantable biosensor that continuously monitors troponin levels after first heart attack to first monitor recovery and second, detect another heart attack occurrence, as there is a 20% chance of this happening again.
So a biosensor is a device that measures the biological concentrations within its environment and generates understandable signals proportional to concentration. A typical biosensor is composed of 4 major components: the analyte, bioreceptor, transducer, and electronics. A notable example of a novel implantable biosensor is the Eversense continuous glucose monitoring system. This device was the first FDA-approved CGM system to include a fully implantable sensor for detecting glucose. Our proposed device will take a lot of inspiration from the Eversense CGM system. The shape will be a small capsule-like design, with the length being around 18 millimeters and diameter being around 3 to 4 millimeters. The purpose of the capsule-like design is that it reduces the amount of rough and sharp edges of the device, which could easily damage the surrounding tissues and produce chronic stress into the nearby tissue. Our proposed biosensor will be encapsulated in polymethyl methacrylate, PMMA. PMMA is a biocompatible, but lightweight and synthetic polymer that has great mechanical properties and low toxicity. Additionally, our proposed sensor would also include a silicone elastomer matrix containing a small amount of dimethicone acetate, which would be used as an anti-inflammatory agent. For the detection of the analyte, the bioreceptor would be an immobilized 76-base aptamer specifically designed to detect the troponin in the range of 0.03 to 2 nanograms per milliliter. Aptamers are synthetic single-stranded DNA or RNA molecules that bind to a wide range of targets with high specificity and affinity. Aptamers would be ideal as troponin biosensor studies have shown that they have a stable, regenerative, and reproducible function. Current studies have shown that the most optimal approach to power implantable biosensors is through wireless technology, as it provides a high rate of data transmission and high level power transmission without the need of a battery. The ideal site to implant this biosensor would be under the skin of the upper arm region, as troponin originates in the cytoplasm of the cardiac myocytes, eventually reaching the interstitial fluids. Next up is the transmitter sensor transmitter mechanism. Troponin-based aptamer utilizes the redox marker ferrinocyanide and gold transducer surface to form a potential gradient that can be quantitatively measured as a change in voltage. This decreases the voltammetric signal produced by the aptamer, which can be measured quantitatively. Once the electrical signal is generated from the aptamer, the signal will go through a built-in transceiver unit to transmit the signal using a specific radio frequency to the transmitter, which is placed over the skin. The sensor will also utilize a different radio frequency on the same chip to wirelessly be powered by the transmitter simultaneously. Our proposed device would use an amplifier in the transmitter to amplify the signal received and pass through high and low pass filters to reduce any unwanted noise from the raw signal. This filter signal then passes through the microcontroller with a programmed signal analysis algorithm. These microcontrollers would then control LED or vibration module that turns on based on the data received in the biosensor to indicate if the troponin levels are higher than the elevated rate in the patient's body.
As the device is implanted subcutaneously in the upper arm region of the body, the device will be in contact with a variety of different analytes. The sensor should function as troponin being the only analyte that binds to the biosensor. The biosensor must also be used at 37 degrees Celsius in order to support the body's temperature sensitivity to provide accurate signals for detection in time-efficient manner. The device must be utilized at an appropriate dosage of dexamethasone acetate to minimize the body's inflammatory reaction to the implanted sensor. The device must be continuous monitoring of troponin levels in the body for up to 90 days. Design objectives allow for biosensors to have sensitivity towards the body such that it must not be chemically conformed to be degraded into the body or have toxicity and inflammation in the body. The device must also be biocompatible and time efficient with caution alerts, and reproducible results must be issued from the device.
From studies about mechanical characteristics of subcutaneous tissue, it is shown that the tensile modulus ranges from 1.6 to 2.9 kilopascals, while the viscosity of the interstitial fluid in the tissue is 36.8 megapascals per second at body temperature. In terms of design specification, the PMMA casing of the biosensor must be able to integrate well with the subcutaneous tissue located near this region, therefore must be properly designed with these values in mind. For reducing inflammatory response, 1.75 milligrams of G-mexithone acetate will be applied to the eluting silicone collar surrounding the sensor. Previous studies done by Eversense showed that this specific dosage was ideal for the most effective anti-inflammatory response. In terms of the design specifications for the sensor transmitter mechanism, the aptamer concentration used for the proposed biosensor is 10 microliters of the solution. The literature also showed that the troponin-based aptamer remains stable for at least 25 days. To reach the functional requirement of 90 plus days, inexpensive chemical modifications have shown to drastically increase the stability of the aptamer, therefore increasing the lifespan. An optimal frequency range for the data power and power transmission must be also considered for the chip to transmit data and power the sensor. Therefore, the frequency band for the data transmission will be following the medical implant communication system med radio frequency band. For wireless power transmission, the frequency band is in the low gigahertz range and has been identified as the ideal range to power the sensor as it provides minimal power loss due to tissue absorption. Finally, to avoid tissue damage, the radiation levels emitted by the radio frequencies must be considered when designing the electrical components of the biosensor. We can use specific absorption rate as a tool to quantitatively measure the power absorbed by the biological tissue. The FCC regulates this as a maximum value of 1.6 watts per kilogram averaged over 1 gram of tissue.
Essential benefits of the proposed device include a faster and more manageable diagnosis. The overall outcome of the proposed biosensor involves a final display that summarizes all findings from the signal processing algorithms for a faster and more manageable diagnosis from home. The fast and speed of the device's detection supports and brings awareness of the patient's severity of heart complications. This is also convenient and cost-saving for a patient, especially non-mobile elderly, because the amount of hospital visits would be reduced greatly, specifically by 38%. The implantable biosensor possesses a great deal of worth to healthcare providers and patients, being that the continuous monitoring of troponin levels is performed, which is necessary and beneficial for other similar heart-based abnormalities, customizing further medicine and treatment for the patient. Alongside the continuous monitoring, daily data on patients' physiology is received by healthcare providers and alert systems to ensure that there is no significant variation in standard values or miss. Some occasional limitations that must be advised are implant migration, as well as the difficulty in obtaining long-term continuous troponin monitoring from the biosensor in a one-period insertion due to the lifespan and gradual sensor functionality loss and stability of the biosensor. Although expected lifespan of the device is 90 days, slight sensor failure in function can occur during this period. Here's the component cost. This table lists all components needed and their cost production in the biosensor. The proposed biosensor closely resembles the design specifications and standards of the Eversense system. Thus, the costs of the systems are similar. The total cost of the proposed biosensor is estimated to be $6,400 per year for each biosensor, or $533 a month without insurance factored in. Additionally, the cost for removal and insertion of the biosensor estimates from $300 to $400, but is determined by surgical healthcare providers.
So after implantation, foreign body response is unavoidable as it's needed to heal. However, we can control it by the right use of materials. We chose biocompatible materials that will prevent fibrous encapsulation, as troponin needs to bind to the gall aptamer, and a layer of tissue around the sensor would stop this process. The encasement is made out of BME, which promotes tissue regeneration, as the studies have shown that BME surface has absorption properties for key basal lamina proteins, and it also inhibits the presence of macrophages. For the aptamer binding surface, we chose gold, as it's one of the most biocompatible metals and has the ability to conduct electrical signals into the circuit. Gold is also non-corrosive and has anti-inflammatory capabilities that prevents slow cell adhesion, which will prevent fibrous encapsulation. Finally, there is the silicone collar that releases dexamethasone acetate. Silicone is an elastomer with drug delivery capabilities. The decision to add this drug was made to control the immune response, as DXA has the capability to suppress inflammatory genes. Drug release will be controlled to be less than 3 microliters per day to prevent adverse reactions.
The manufacturing process for the device is designed such that it can be purchased with the insertion tools included in packaging. No personalized fabrication of the device is required, as the size and weight are feasible and suitable for all patients. Most of the weight of the device will be contributed from the power source and encapsulation components. The manufacturing process also includes the device being produced with constant sizing and technical abilities for its specific purpose. From the holder, the sensor and insertion tools will be manufactured and provided sterilized for single use prior to implantation via ethylene oxide for healthcare professional use. The recommended temperature of storage for sterilized biosensor is 36 to 46 degrees Fahrenheit with expiration after 1 month. In addition, the provided ethylene oxide insertion tools are recommended to be stored at room temperature, expiring after 6 months.
Validation and testing. To admit the product as a functional one, several mechanical tests should be performed, starting with basic functionality tests, which would be done to approve selectivity, sensitivity, accuracy of the aptamer to the specified muscle protein, troponin. Another mechanical test would be thermal shock, where the device should work normally after being placed in water bath, heated rapidly from low 5 degrees Celsius to 35 degrees Celsius. It is important to assure, though, that the processed sensor does not cause any potential biological risk to the patients. Thus, multiple biocompatible tests like genotoxicity, cytotoxicity, and hemocompatibility. To continue with the electrical testing, this is essential to admit the device as safe to operate inside the body. First, we will start with testing the general device safety, ensuring the device will operate under specific electrical conditions, such as the signal from the transmitter to perform its intended function. This will ensure that the inductive coil and the transducer will not cause any harm inside the body. EMC testing will be performed as well for any electrical and electromagnetic signaling interference to or from the device. The in vivo tests, small models will be performed on rats for anti-inflammatory agent tests, and then large models will be porcine, young porcine. The choice was related to same skin structure and heart function. We can take any questions at this time.